This recorded session is dedicated for the completion of our module number 4 on biochemistry and physiology of microbes. So I will be discussing the remaining slides on cell physiology and metabolism. I think I have already uploaded the PDF copy of the slide deck, so it will be useful if before you listen to this recorded session, you have the PDF material with you. Okay, so now that we have discussed all the necessary um, all the prior topics about cell motility, also the what they call this the structure of cells, the structures of cells needed to make sure that cells associate with one another for the case of biofilm formation. Also, we made mention about nutrient storage, how cells um, facilitate the reservation of materials from the environment, chemicals from the environment, how microbial cells ensure that they have stashes of these elements. And we also mentioned about, or we also discussed about uh, the capacity of microbial cells to resist a hostile environment by forming resistive structures like spores and other related microscopic I don't know microscopic structures like spores for bacteria and um, endospores for soil and actinobacteria. Okay, all those prior topics are leading us to the main story for this module, which is the how the environment interplays with microbial response in order to ensure that a particular physiological type of microbe will only exist on the specific area. So the idea of selective growth response. I'd like you to think that, or I'd like to impart how I think or how I believe that it's both the microbe in its goal or with its intention to survive, to reproduce, and the environment with its provision of physical, chemical, and biochemical parameters. How these two things interplay in order to dictate what kind of microbe will be found on a specific environment, on a specific area of the environment. To give you um, some illustrations. Let's have the example of the microbial mat. Diba? So in a microbial mat, the top layer, which is frequently exposed to sun, to exposed to water as well, running water, we expect that photosynthetic bacteria will dominate on the top layer of the microbial mat because everything they need is in abundance on that location. The other hand, if you, let's say, have a confined environment where organic matter is very high, not much, not much presence of light or gases, we may expect that fermentative bacteria will dominate. Okay, unlike the other types. So, for a specific group of microbe to dominate in such various conditions, they must have appropriate sensory response systems. They must be capable of regulating their DNA expression, and they have defined structures, of course. Like for the case of bacteria that have very resistant cell membrane and cell wall for aquatic environments where salt concentration is very high. That's one illustrative example. And definitely, bacteria must be capable of regulating and producing enzymes that are important for them to carry out their life processes. Let's further talk about sensory response and global regulation. So these two topics are the highlight of our module. No? So in nature, stimuli may be of different types, may be of different forms, no? Such as chemicals. For chemical stimuli, we have compounds that are released by plants, by animals, or other microbes, other bacteria. And they can influence how a bacteria will move from one location to another if it's trying to avoid that stimuli. Let's say the stimuli is a non-beneficial one, let's say a toxin from the plant that might kill the bacteria or the microbe, then the microbe will be influenced to, or will be dictated by its sensory system to move away from that toxin. Diba? In some instances, bacterial movement may also be positive, in which if the release stimuli or the stimuli is a beneficial one, let's say it indicates the richness of a nutrient or a gas, no? It can attract the microbe to go to that location. No? So these chemical stimuli not only affects bacterial movement, microbial movement, but also metabolic processes and development of cell structures like endospore formation. So you remember that in hostile conditions, water very low concentration, very dry surroundings, temperature is harsh or temperature is freezing. Some bacteria, like the soil inhabiting ones, are induced to produce endospores in order for them to survive. No? And aside from chemical stimuli, we also have physical stimuli, like for the case of osmotic pressure. And this physical stimuli, set of physical stimuli, may elicit changes in the bacteria. Also in the same departments. Could be movement, affecting metabolism, or affecting overall bacterial regulation by the development of adaptive cell structures. No? In the context of microbial communities or mixed species growing in a particular space where the stimulus is applied, it is natural for us to expect that not all of them, not all of these species that are cohabiting with one another will respond to the stimulus in the same degree. No? Or you may also think of it na not all will really respond at all. No, well, not all species will or some species may not respond to the stimuli because simply this simply relies on the fact that not all bacterial species will have the same sensory systems. Okay, so in different species that possess the necessary sensory system to sense a particular stimuli, different responses may also be expected. Like for the case of osmotic pressure. No? So for the case of osmotic pressure, increase in osmotic pressure can be sensed by bacteria like vibrioids. Like for vibrioids, they will do toxin production. They will respond by producing toxin if the osmotic pressure of the environment becomes high. For the case of gamma proteobacteria like Pseudomonas, so Pseudomonas may induce the production of capsule formation in order to prevent leakage of the cell because of the sudden change in the osmotic pressure in the environment. And then for the case of other bacteria like Shigella, no, increase in osmotic pressure will mean a different response. As unlike Pseudomonas or Vibrio, for Shigella, synthesis of porine materials, porine proteins, and their placement in the cell membrane will be the response of Shigella species in order to counteract the effect of an increase in the osmotic pressure. So truly, this different stimuli will have different effects to our microbial communities or microbial species in the environment in nature. No? Sensory systems that are used by microbes to respond to changes in the environment with the cellular induced effects, okay, will essentially result to selective adaptation or selective growth. No? While there are different types of sensory responses in bacteria, two that I will give emphasis for this lecture are two component systems, mga TCS, and quorum sensing systems, QS systems. Okay, so let's first talk about two-component systems. Two-component systems are the most simple type of sensory systems in microbes and bacteria, where the transduction of signal from the environment to the inside of the microbe is facilitated by a set of proteins, no? A set of membrane proteins. So what happens is initially there is an interaction between your sensory protein and the stimulus, and then that interaction will actually result to a conformational change in the sensory protein. No? The phosphorylated sensory protein will become dephosphorylated, okay, and in the process, it phosphorylates the receptor protein, the cytoplasmic receptor protein. No? So once that there is a transfer of the phosphate group to the cytoplasmic receptor protein, the phosphorylated receptor protein is now capable of interacting with the DNA itself. Okay, and may act as a repressor or a promoter, such that DNA expression of a particular protein will be regulated. No, so that's the basic discussion of what two component systems are. Okay, so you have your sensory protein and your receptor protein. Kaya nga siya tinawag na two component, because only these two are involved for the heavy activities. However, we have to also what do you call this? We also have to remember that within two component systems, no, a more complex concern or more complex narrative is how or what the quantity of response or components that are actually regulated by the whole process itself. Because it's possible that or not really possible, but in reality, okay, um, one regulator protein will not be a dedicated protein to a single function. So you have to remember that. Huh? A receptor protein will once activated may induce branching effects or branching effects or branching activation of multiple components, be it in the DNA, like in order to regulate gene expression of multiple gene expression, or could also be as an interacting or as an active molecule that can activate other molecules that will be involved in multiple functions and multiple responses. So, for example, um, we have a phosphorylated receptor protein that can coordinate positive chemotaxis towards a sugar source, and at the same time it can interact with a sugar transport system in order to respond in order to produce an immediate bacterial response. So in reality, that's what happens. There are complex branching, complex mechanisms or and routes of signal transduction. It's not really it. The system itself is very straightforward, but you have to remember that in different contexts, complexity will arise. Okay. So now let us take a look at some examples of bacterial sensory systems and the set of stimuli that can activate or that can be perceived by these systems in order to mount a particular response. So we have chemotaxis. Chemotaxis as a system perceives chemical attractants, and as such, they can induce the cell to move using its flagella towards or away from the stimuli. Also, in the case of spore production, a change in the cellular density as well as change in temperature or pH will result to cells ensuring that they need to survive in the following seasons. So some of them will produce spores. Also, we have chemicals that are secreted by plant roots. For the case of rhizobial symbiosis, for the case of nitrogen-fixing bacteria. No? So the stimuli for them to start forming the nodules in bacteria in plant roots is the chemicals that are secreted by plants. So those chemicals are most likely attractants because your nitrogen-fixing bacteria will migrate to that area of plant roots and they will begin invasion and nodule formation. And for osmotic pressure, I've mentioned this as an example. We also have presence of metals at sublethal concentration. Your heavy metal resistant bacterial strains have resistance mechanisms or resistance effectors that are essentially proteins, and you expect that these proteins will not be expressed if your bacterial strain is not exposed to a condition where there's high concentration of heavy metals. Diba? Then we also have other examples. For a case of osmotic pressure, then capsule synthesis. Okay, so these sensory systems are generic examples that occur in different groups, no? In multiple groups of bacteria. Okay. Aside from two-component systems, we also have quorum sensing, which is another communication method among bacteria. However, this type factors in the cell population. So quorum sensing factors in cell population. So we can define quorum sensing as a cell density dependent communication among bacterial cells, which is facilitated by the production and release of extracellular agents known as autoinducers. So what happens in a quorum sensing setup? So the goal is to initiate gene expression if the optimal cell density of the bacterial group is achieved. So how does it happen? Initially, low populations or low numbers of bacteria of a specific species will produce the autoinducer, the chemical at low concentration, and this chemical is exported outside. It's secretory, so it will go outside the cell, and then it will stay in the surrounding environment. Normally, this autoinducers are hard to be metabolized, so they will not really be degraded by other microbes in the environment. Then eventually, when the bacteria of concern continues to divide, it will continue to increase its number. Now, the more autoinducers will be produced because there are more bacterial cells now. The increase in the concentration of autoinducers is inevitable. No? Now, the concentration of that autoinducer, the sensory chemical, will come to a point where will achieve a threshold where it's already sufficiently great so that it can now go back, no, to the inside of the cells, because I remember they are secretory. So the natural concentration gradient is that as the bacteria producing them will increase in population, in numbers, the more that they will be produced and will be secreted outside. Now that there's high concentration of these autoinducers in the environment, it's now time for the autoinducers to go back to the bacterial cells and do their thing, which is to facilitate DNA expression. So these autoinducers may serve as promoters or repressors, and as such, the process of quorum sensing is really a unique one in the sense that it will only produce something needed if there is enough population of cells that will make use of it. So para we factor in we factor the idea of microbial economics na if a single bacteria will produce something that it will use, eventually it will be tired and will be exhausted of producing it. So in quorum sensing systems, that kind of inefficiency is avoided. So it's really a good, I don't know, it's a nice, or it's a wonderful natural phenomenon that a lot of bacteria are capable of exhibiting. It's something that you might consider if you once that you start doing your thesis na, quorum sensing systems. Okay. So just a representation. Your quorum sensing system will allow your bacteria to control gene expression in response to the population size. And at threshold concentrations, the autoinducers will activate regulators that will target specific genes, or they themselves may act as regulators. Right. Some examples of autoinducers that are used by microbes to facilitate cell-to-cell communication. The most popular one is represented here in Figure A, acyl homoserine lactone, mostly for gram negatives. So you expect quorum sensing gram negatives to make use of AHL as their primary autoinducer. Now we also have another example, hydroxypalmitic acid methyl ester, which we associate with Ralstonia genus. And then for a yeast, we have farnesoic acid as an example of autoinducer. Okay, so the others, I will leave to you if you want to further read about them. Okay, so we're done with sensory systems. No? We have QS and we have TCS, quorum sensing, and we have two components. Now we will finish our discussion with global regulation in prokaryotes. No? DNA expression is a costly activity, and therefore it must be regulated to ensure efficiency. Only proteins that are needed in the specific environment where the microbe is thriving must be produced at the time that they are needed. So, some enzymes required for basic synthesis of cellular structures and for metabolism are produced continuously, and we refer to that production as constitutive protein synthesis. So there are cellular subcomponents that are important because of the bulk that the abundance that they have in the cell. So they must be produced and replenished from time to time. They undergo what we call constitutive protein synthesis because there should be no exception in producing them. Parang walang dapat na walang dapat na trigger needed anymore because the fact it's abundantly used, it's abundantly present in the bacterial cell, abundantly utilized in many processes. That fact alone, dictates that the bacteria must produce these constitutive or must produce these proteins in a constitutive manner. On the other hand, we also have those that are produced in an inducible manner. So those inducible proteins, usually the things that they process, the substrates that they break down, the components that or the structures that they are components of, those structures and substrates are very rare, no? Kaya siya inducible because there will be special occasions that those substrates and those structures are formed. Therefore, the bacteria, the prokaryote, does not need to produce the proteins necessary or in complementation of those structures and those activities. So those proteins are produced in an inducible manner. Okay, so there is tight regulation in order to produce such proteins, no? Ensuring that there is efficient coordination of gene expression, di ba, among inducible operons. Ano pala, no? Let's go back to some definition on operons, di ba? So, from your genetics, you remember that operons are clusters of genes that do a common thing, do a common function, and they are usually repressible or inducible, no? So, in cases where the cell need to produce an inducible protein, an inducible need to carry out inducible protein synthesis. Most of the time, multiple operons are involved, and in of course with the concept of microbial economics, the cell needs to ensure that the operons that are involved in the process of producing the components, the component proteins, they should be tightly regulated. That they should be active at the same time when they are needed, or they should be however, in some cases, about one operon might deactivate another in order to carry out with the succeeding steps. Now that's another variation of the process. But in summary, or a key thing that I want you to remember is in this tight or in the phenomenon of tightly regulating gene expression, no, um, um, your what do you call this? Your cell will have certain effectors, no? So they will have certain effectors, especially most likely proteins to, yung mga effector na to. We call those proteins, regulons. No? So regulons will uh, later I will later on the presentation I have an example, but to illustrate it na. No? So regulons are um, um, major proteins, major in the sense that they affect multiple signal transduction pathways. They affect multiple uh, protein expression pathways. No? So later I will provide a very good example of what a regulon is. Now I want to really reinforce the idea of tight global regulation in microbes. So I am having this table here. I presented this table here. Let's have some examples of or let's take a look rather at what do you call this at certain molecules and activities and the percent composition that they have in our cells, as well as the percent of total biosynthetic energy required to synthesize them. And as you can see, proteins are the number one. Sila yung top one. No? Almost half of the energy needed or energy produced by a cell, by a microbial cell, is dedicated. Actually, more than half, 52% of the biosynthetic energy um, produced by a cell at a given point in time will be dedicated, will be diverted to protein production, to gene regulation for protein expression, and the others will follow. Polysaccharides, RNA, DNA actually has a very less requirement in terms of energy. Then for cell composition, we can relate that the more a biomolecule or a process is present in a microbial cell, the more energy required to produce them, the more time dedicated by the cell to regulate the process of producing them. Okay. Now let's have some examples of globally regulated processes in bacterial systems. So these are things that I have been mentioning since module one. So respiration, repression, DNA repair, oxidative stress, nitrogen utilization. And if you're going to take a look at this table, one thing that you will be able to infer is that um, stimuli responses are requiring less genes compared to induction or repression processes. Diba? So for example, respiration will only require more than 50 or 70 genes involved. Then for catabolite repression, okay, we have more than 300 needed. So the regulatory processes, we can say, in as a rule of thumb, we expect that regulatory processes will require more genes, no? Will require the involvement of hundreds to thousands of genes, no? Hundreds of operons in order to ensure the smooth execution of the regulatory process. While the responses, hindi naman ganoon karami ang genes involved. But I don't think that konti yung 70 genes for the case of respiration, or 36 genes for the case of heat shock response. That's already too much, diba? Considering the relative size of a microbial prokaryote compare sa atin, multicellular animals. Diba? You just think of how grand the regulated processes are, even though in this very minute, very minute living systems. Now, as promised, I will be discussing a specific case of regulon. No? So, in the bacteria Myxococcus santus, which is a predatory microbe. No? Starvation triggers the formation of multicellular fruiting bodies that contain thousands of resistant spores, no? Stress-resistant spores. And current studies on this bacteria identified a major regulon, the protein NLA6. It is defined, or it is identified as an enhancer binding protein. So as a regulatory protein, as a regulon itself, it's capable of regulating different activities of certain genes dedicated for growth, for spore formation in order to, I don't know, here for spore formation, and also transcriptional regulation of other necessary genes. No? So as a regulon, no, an enhancer binding protein, it can bind directly to promoter regions of the whole operon on each of these functions, no? So that the processes will be carried out or will be activated or repressed at a certain time. No? This paper discusses further on how NLA6 actually becomes a or is identified as a key component in the regulation of transition between a vegetative state of the Myxococcus santos model to a resistant, no, to a sexual state, yung ano siya, yung nag-transition, yung pag-transition from a vegetative cell to the production of spores. NLA6 is a key regulon. No? You may want to view or read the whole article from this inset. I've included the link here. It's open access, so you can access it without any subscription. Now, one of the data that they have to suggest or to substantiate yung assumptions that I've been mentioning, which are based on the paper, is they develop phenotype strains that carry mutations in the NLA6 genes. No? So the NLA6 gene that produces NLA6 protein, so mutate nila yan. And then what they saw after reproduction and after the whole cycle, the viability of the spores decrease. If you're going to compare viable number of spores compared to the wild type, the one that does not have any mutation on the gene producing the regulon, wala. Sobrang liit ng viable spores produced. So that's one level, no? First level of seeing the effect of the mutation. Another level or a secondary level is they produce mutations in the target genes, okay, of NLA6, 2688 and 3259. And then after producing those, I think insertions yung mga ginawa nila, no? Nag-insert sila ng mga sequences to mutate the promoter region. No? So for the cases of strains that have mutations on the target genes but not on the regulatory protein, the same observation also is obvious or has been recorded, that the viability of the spores decrease. So suggesting that indeed this protein here, NLA6, no, as an enhancer binding protein, is a master regulator, not of itself alone, but also of other processes involved in the transmission, or rather in the transition between vegetative state to a dormant state. No? This is a very nice paper showing us a lot of, actually if you're going to read through it, it contains a lot of data really substantiating the claim. So really believable. And I think talagang there's enough credence that we should be giving to works like this. Now, too much about this too much about discussion on regulon. Now, at this point, I can no longer express the absoluteness of the fact that microbial growth is dependent on electron flow. Now, electron flow through a series of redox reactions and microorganisms having the capacity to obtain energy from various inorganic electron donors or electron acceptors. So all life, microbial life, and even our lives as multicellular organisms depend on this curated and tightly regulated process of electron flow. No? So for microbial growth, ATP must be produced. And major mechanisms for producing ATP in biological systems include what we already know so far: oxidative phosphorylation, substrate level phosphorylation, and photophosphorylation. Probably you have encountered these terms in photosynthesis, in glycolysis, in Krebs cycle, diba? So as a review, we have oxidative phosphorylation as a process in which ATP is produced as a result of electron transfer from donors such as NADH or FADH to oxygen. Kaya nga siya oxidative phosphorylation. No? So the process normally takes place or is expected to be happening all the time in the mitochondria of eukaryotic cells. And for aerobic microorganisms or aerobic organisms like us, oxidative phosphorylation is a major source of ATP. And then we also have substrate level phosphorylation. We relate to glucose breakdown, glycolysis. So it refers to the formation of ATP from ADP and a phosphorylated intermediate rather than ADP and inorganic phosphate. No, that which is actually opposite of oxidative, which is actually what's happening in oxidative phosphorylation. So in substrate level phosphorylation, there is a phosphorylated intermediate that serves as the source of the phosphate group, no, for ATP production from ADP. Then lastly, we have photophosphorylation, which is the process of utilizing light energy from photosynthesis to convert ADP to ATP. It is a process of synthesizing ATP molecules by transferring the phosphate group into ADP, while light is in the background or light is involved. No? Light energy is involved. We discussed a number of metabolic categories of microbes, and in one of the modules that we have, no, in lab, yung ating, I think module number 3 natin, no, for soil microbes, no? Soil and air microbes. So two groups that we will make a quick mention again are the heterotrophs and the chemolithotrophs. In terms of distribution, our heterotrophs are the widely dispersed microbial groups in our planet. So the reason why, or not really the reason, but the characteristic that they all have is they make use of pre-made compounds. They already make use of they make use of already available compounds in the environment. Compounds normally synthesized by other microbes, by plants or animals. No? And most heterotrophs have the capacity to make use of glucose because glucose is the most abundant sugar in the planet. No? For heterotrophs to diversify, they need to have life adaptations to make use of glucose. No? So most heterotrophs have the enzymatic capacity to utilize glucose. Lastly, let's review chemolithotrophs. So chemolithotrophs are microbes that use inorganic compounds or minerals either as their electron donor or their electron acceptor. For the case of those that make use of carbon dioxide as their carbon source, the microorganisms are known as chemoautotrophs. So most of the microbes that we will be discussing moving forward are either heterotrophic or chemolithotrophic. So most of them that we will be mentioning in our succeeding modules. That's why it's important to really emphasize on these two groups. No? Now, some key terms for your review, I have listed here some terms on cellular energetics. I want you to review this on your own kasi these are fundamental bio concepts. So I don't have to dedicate an extra time to mention them in our lecture. So for us to, or for you to be able to follow our discussion, then please review these processes, these cellular energetics processes. Glycolysis, the TCA cycle, fermentation, electron transport, then the three types of phosphorylation which I mentioned a while ago. Okay, so that this concludes our lecture for module number 4 on the biochemistry and physiology of microbes. A few synthesis questions for your review also I have here. These questions are lifted from Barton, so if you want to check out the ebook, then you will be able to see the same synthesis questions. Okay, so this ends our session for this module, for module number 4. I will be uploading this one in YouTube and we'll make the link available.